Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be going over regulation of cholesterol biosynthesis. So all the types of regulation over cholesterol biosynthesis in our cells will be explained in this lecture. Now let's get into the details. Regulation of cholesterol biosynthesis can be explained in under following headings. First we will go over feedback regulation, and then I will explain you covalent modification, and then the transcriptional control of cholesterol biosynthesis, and the regulation with proteolytic degradation of HMG-CoA reductase, cholesterol balance in tissues, and LDL receptor regulation, and at the end we will go over some of the drugs that are inhibiting cholesterol biosynthesis. So these are the titles under which we will understand how cholesterol biosynthesis is regulated in our cells. So the first thing is feedback regulation. Now the most regulated enzyme in cholesterol biosynthesis is HMG-CoA reductase enzyme, and this HMG-CoA reductase enzyme, like any other regulated enzyme in a metabolic pathway, will have a feedback effect from the end product of the pathway, and the end product of the pathway in cholesterol biosynthesis, as you know, it is cholesterol. So whenever cholesterol is accumulated in the cell, this cholesterol will have a negative effect on HMG-CoA reductase enzyme. Not only that, the immediate end product of HMG-CoA reductase is mevalonate, because HMG-CoA reductase is going to convert HMG-CoA into mevalonate. So whenever there is a buildup of mevalonate, this mevalonate will have a negative effect on HMG-CoA reductase enzyme. And also note that whenever we consume excess cholesterol in the diet, which is being absorbed by our intestine and transported to liver and other tissues, mainly to the liver, so that dietary cholesterol will also have a negative effect on HMG-CoA reductase, especially in the hepatocytes, because most of the cholesterol that is absorbed from the intestine will be entering into the blood in the form of chylomicrons. Chylomicrons undergoes metabolism in the circulation into chylomicron remnants, where only triglycerides are taken out, but the dietary cholesterol will reach liver in the form of chylomicron remnant, and that dietary cholesterol in the liver, it can regulate HMG-CoA reductase enzyme activity. That's how dietary cholesterol will have a feedback negative effect on HMG-CoA reductase enzyme. Now, consumption of about 100 mg of dietary cholesterol is equivalent to decrease in approximately about 0.13 millimoles of cholesterol in the serum. So that's how the calculation can be done when we advise a person to decrease cholesterol levels in their diet. Coming to feedback regulation on HMG-CoA reductase using the figure here for the better understanding. So the immediate end product of HMG-CoA reductase is mevalonate, and that mevalonate will have a negative effect on HMG-CoA reductase, and the cholesterol, which is an end product, has got a negative effect, and the bile acid, which is a derivative of cholesterol, also have a negative feedback effect on HMG-CoA reductase enzyme. And here in the figure, as it is shown, the statins, which are the drugs that act on HMG-CoA reductase competitively, they are going to compete with HMG-CoA for the binding site. So they will also inhibit HMG-CoA reductase enzyme, thereby reduce formation of mevalonate, and ultimately will decrease cholesterol biosynthesis. Now let's move on to see other type of regulation of cholesterol biosynthesis, and that is covalent modification. What happens here? First thing we should know that the enzyme HMG-CoA reductase, it will be active in dephosphorylated state. Okay. So whenever a enzyme is dephosphorylated, done by insulin and thyroid hormones, so the dephosphorylated form of HMG-CoA reductase, it is an active form of an enzyme. And also note that most of the biosynthetic enzymes, the regulated enzymes in metabolic pathways which are regulated by insulin, they are active in dephosphorylated condition. Remember that. Insulin, whenever it is promoting a metabolic pathway, this is general point that you should remember. Whenever insulin is promoting a metabolic pathway, usually that keeps enzyme in a dephosphorylated state, especially the most regulated enzyme. Okay, so in the same way, here cholesterol biosynthesis, HMG-CoA reductase is the most regulated enzyme, and insulin, which is promoting biosynthesis of cholesterol, it is going to keep HMG-CoA reductase in dephosphorylated state, and that's an active state of an enzyme. And insulin always works through protein phosphatase to remove phosphate from the surface of an enzyme. Thyroid hormones also keep HMG-CoA reductase in dephosphorylated state and keep the enzyme active. Whereas the antagonistic hormones like fasting hormone glucagon or the stress hormone like glucocorticoids, right? So they are going to phosphorylate HMG-CoA reductase, thereby keep enzyme inactive. And when a muscle contraction is going on, like during exercise, as you know, ATP breaks into AMP. ATP to AMP is like ATP to ADP, ADP to AMP. And that AMP is going to activate protein kinase, AMP activated protein kinase, that is AMPK. And this AMP activated protein kinase is going to phosphorylate HMG-CoA reductase and keep it inactive. Okay, and that is all shown here in this particular figure. As you see, insulin is going to activate protein phosphatase enzyme and maintain HMG-CoA reductase and in the active condition. Thereby, HMG-CoA is converted to mevalonate, and thereby cholesterol biosynthesis is increased. Similarly, thyroid hormones also does the same thing. And glucocorticoids, glucagon, these molecules, they are going to phosphorylate HMG-CoA reductase enzyme, thereby they keep the enzyme inactive. Similarly, AMP via AMP activated protein kinase also keep HMG-CoA reductase inactive by phosphorylating it. So that's the covalent modification there. Now coming to proteolysis of existing enzyme, that is HMG-CoA reductase enzyme, as it is shown here in the figure B. Now HMG-CoA reductase enzyme, it is embedded in the endoplasmic reticular membrane, and it has got a cytoplasmic domain, and that cytoplasmic domain has a cholesterol sensing regions. And whenever there is excess sterol, means cholesterol, it will go and bind to this cholesterol sensing domain of HMG-CoA reductase, and it is going to change the conformation of an enzyme, thereby it is moving into proteolysis or protein degradation, thereby enzyme levels decrease. That's another way of controlling or regulating cholesterol biosynthesis in our cells. Now let's move on to learn how a cholesterol biosynthesis is controlled at the transcriptional level. You know, the most important enzyme in the cholesterol biosynthesis pathway, which is most regulated, is HMG-CoA reductase enzyme. Now in the transcriptional control or regulation, so the regulation will be going on over the gene that is coding for HMG-CoA reductase enzyme. That means the messenger RNA that is coming out of the gene. So more messenger RNA coming out means more translation will go on and more HMG-CoA reductase enzyme will be synthesized. More HMG-CoA reductase means more cholesterol biosynthesis, right? So how that is done? So SREBP is a sterol regulatory element binding protein. It has got an N-terminal DNA binding domain as it is shown here in the figure. And this SREBP, it is a secretory protein, so that's why it is synthesized. It is there over the endoplasmic reticular membrane, ready to move on to Golgi complex. And in the Golgi complex, we have S1P and S2P, that is site 1 protease and site 2 protease. They are going to clip that DNA binding domain and allow that DNA binding domain to go into the nucleus, and in the nucleus, DNA binding domain will go and bind to a sterol response element or sterol regulatory element, which is a part of the gene segment in the upstream of the gene that is coding for HMG-CoA reductase enzyme. And when SRE binds to SREBP DNA binding domain, at that time gene transcription will go on and mRNA for HMG-CoA reductase enzyme will come out, and that will undergo translation to make HMG-CoA reductase enzyme. That's how the process goes. And also note that this DNA binding domain of SREBP, so it is a short-lived molecule. That means it will be destroyed, degraded often. So that means every time that transcription has to go on, so new DNA binding domain has to come, so that a new HMG-CoA reductase enzyme transcription may go on, right? This is the normal process. Now how cholesterol is going to control this? So let's see when a person has got higher cholesterol level. When the cell has got higher cholesterol levels, that cholesterol will go and bind to cholesterol sensing domain, which is present over S-cap. As it is shown, there is a protein called S-cap, and that S-cap has got a cholesterol sensing domain. So cholesterol, whenever it is at higher levels in the cell, will go and bind to cholesterol sensing domain of S-cap, and S-cap makes an association with SREBP at that time. Now the SREBP S-cap, it do not move on to the Golgi because cholesterol is attached. So whenever cholesterol is binding to S-cap, SREBP S-cap complex do not move to Golgi. And if it do not move to Golgi, then DNA binding domain is not released. If the DNA binding domain is not released, that DNA binding domain, which is a SREBP, sterol response element binding proteins domain, is not going to interact with SRE, that's a gene segment in the upstream elements, right? So mRNA for HMG-CoA reductase enzyme is not coming out. That means HMG-CoA reductase enzyme levels will decrease. That's how higher cholesterol level in the cell will decrease the transcription of the gene that is coding for HMG-CoA reductase enzyme, thereby decrease the synthesis of HMG-CoA reductase, that will decrease the synthesis of cholesterol. Now let's look at the opposite condition. Whenever cholesterol levels are low in the cell, what will happen? So cholesterol do not bind with S-cap. So when the cholesterol do not bind with S-cap, now the S-cap, it can move, which is associated with SREBP, it will move on to Golgi membrane because cholesterol is no longer binding to S-cap because it is low, cholesterol levels are low. So because of this, what happens? S-cap SREBP, because it moved to the Golgi, in the Golgi membrane we have S1P and S2P. S1P and S2P, these are proteases, right? So they are going to clip that DNA binding domain, and DNA binding domain will go and bind to SRE, and the gene transcription will go on, and you get mRNA for HMG-CoA reductase enzyme. That means there will be synthesis of HMG-CoA reductase, and when there is a synthesis of HMG-CoA reductase, concentration increase means cholesterol synthesis will go on. So, and when the more and more cholesterol is synthesized, then that cholesterol will go and bind to S-cap and do not allow S-cap SREBP to move on. That means DNA binding domain do not go and bind, so transcription will go down, right? This is how the levels of cholesterol inside the cell will control the synthesis of HMG-CoA reductase. This is the transcriptional control. Now let's move on. So whenever we have higher levels of cholesterol, this cholesterol will go and bind to cholesterol sensing domain of HMG-CoA reductase enzyme, which is there in the endoplasmic reticular membrane with the cytoplasmic domain, and thereby the conformational change in this particular enzyme because of the binding of cholesterol molecule to the sensing domain, cholesterol sensing domain, will predispose to this particular enzyme after the conformational change to a proteolytic degradation. Thereby, enzyme levels will go down. Next, move on. Now, how to balance cholesterol levels in the tissue? Now, the cholesterol levels in the tissue is balanced in following ways. Intracellular cholesterol levels regulate LDL receptor synthesis and expression of LDL receptor synthesis over the membrane. Note that whenever there is a low levels of cholesterol inside the cell, then there will be increase in the expression of LDL receptors over the membrane. So it's a reciprocal relationship that you should remember. Lower the cholesterol in the cell, higher the LDL receptors over the membrane. And that makes sense. Why? Because LDL is a cholesterol rich molecule. When the cell has less cholesterol, so LDL has to be taken into the cell so that cell gets cholesterol, right? So that's why LDL receptor responds to intracellular cholesterol level. How that is done? So the cholesterol itself, it will have a transcriptional control over the gene that is coding for LDL receptor. So whenever a lower cholesterol is there inside the cell, and that cholesterol will increase the transcription of LDL receptor by a SREBP mechanism. Thereby LDL receptor expression over the cell, cellular membrane increases. And if there is more cholesterol inside the cell, expression of LDL receptors will be decreased. So availability of LDL receptor over the cell membrane will also decrease. Not only that, tissues can also take up cholesterol via non-regulated scavenger receptors if there is a need for that cholesterol. It is not just LDL receptor. And also cholesterol levels in the tissue can also be decreased by efflux of cholesterol outside the cell into the HDL via ATP binding cassette transporters like ABC-A1 transporter, ABC-G1 transporter, and also scavenger receptor B1 in the extrahepatic tissues. So any tissue that has excess cholesterol, it will just pump it out into the blood, and in the blood which is taken up by the HDL, cholesterol is taken up by the HDL and convert that into cholesterol ester by an enzyme called 𝗏-𝐾AT enzyme, which is located over the HDL membrane. That's how cholesterol, excess cholesterol in the cell can be pumped out and cholesterol levels in the cell can be regulated. And also whenever there is a more cholesterol inside the cell, some of that cholesterol can be esterified into cholesterol ester and stored there, because cholesterol ester is a storage form of cholesterol. And that particular esterification, it will be done by 𝐴-𝐾AT enzyme in the endoplasmic reticulum, and the 𝐴-𝐾AT is acyl cholesterol acyl transferase. And also note that depending on the cellular type, cholesterol can be used for the membrane formation. It can be used for steroid hormone synthesis like in the adrenal cortex or in the gonads where estrogen and testosterone can be synthesized, and in the liver bile acids can be synthesized. So like this, cholesterol can be used for the metabolic needs of the cell. Our next way of regulating cholesterol is LDL receptor regulation. The LDL receptors that are present over the cell membranes can be regulated depending on the amount of cholesterol that is present in the cell, which I have already explained to you. So when there is more cholesterol inside the cell, so there will be less LDL receptors which are expressed over the cell membrane. And when there is less cholesterol inside the cell, so there is a more LDL receptors expressed over the cell membrane. So it's an reciprocal kind of relationship. Higher the cholesterol inside, lower the LDL receptor over the membrane. Lower the cholesterol inside, higher the expression of LDL receptor over the membrane. And also note that the LDLs, when they bind with LDL receptor over the cell membrane, as it is shown here, which I have highlighted with the yellow color. So once LDL internalized with the LDL receptor, so they will go and fuse with the lysosome to become endolysosome. And in the endolysosome, so because of the acidity there will be change in the hairpin loop conformation. Basically, the hairpin loop conformation will go on in the LDL receptor, which is attached to the LDL, which when it gets into the endolysosome. So because of the hairpin loop conformation, LDL is separated from LDL receptor. Now the LDL receptor is recycled back in over the membrane or the cell membrane to bring in more LDL molecules in. So LDL receptors are recycled. So in the 20-minute lifespan of LDL, note that LDL receptor, it lives for 20 minutes. So in the 20 minutes lifespan, LDL receptor will be recycled every 5 minutes. That's how the timing goes. Okay. So in a 20-minute lifespan, LDL receptor, whether LDL binds it or not, it is internalized and it's kind of recycled every 5 minutes. Okay. Now, that recycling process needs hairpin loop conformation in the endolysosome. There are certain molecules, small molecular weight proteins called PCSK9s. And this PCSK9s, that is propeptidase, convertase, subtilisin, 𝐒𝐻MG type 9. Proprotease, convertase, subtilisin, 𝐒𝐻MG 9 proteins. What they do is they kind of regulate the expression of LDL receptors over the cell membrane by regulating the recycling process. So what they will do is whenever PCSK9s are there, which are expressed in response to the cholesterol levels that are present inside the cytoplasm. So if there is more cholesterol levels inside the cytoplasm, PCSK9s are expressed, and this PCSK9s, what they will do, they will go and bind with the LDL receptor and do not allow that hairpin loop conformation. When the hairpin loop conformation is not done, that means LDL is not separated from LDL receptor and entire thing is degraded by lysosomal enzymes. That means receptors are not recycled. So the point here is PCSK9s do not allow recycling of LDL receptor. Remember that point. PCSK9s do not allow binding of PCSK9 to LDL receptors, do not allow recycling process, thereby decreases the availability of LDL receptors, and that's how cholesterol levels inside the cell can be controlled. Now, in a person who is having hypercholesterolemia, where LDL cholesterol is levels are high in the blood, like LDL levels are high in the blood. Now, one of the way to regulate that is to control that PCSK9 activity. So these PCSK9s can be inhibited by a class of drugs called as PCSK9 inhibitors. What they will do? So the PCSK9 inhibitors are alirocumab, uvalocumab. What they will do? By inhibiting PCSK9, so they will allow recycling of LDL receptors over the cell membrane, thereby more and more LDL receptors are available over the cell membrane, and LDLs can get into the cell and thereby decreasing the LDL levels in the circulation. That's how hypercholesterolemia can be controlled in one way. So this is how LDL receptor number can be regulated, which are present over the cell. Now let's move on to see a regulation of cholesterol biosynthesis by using a class of drugs called HMG-CoA reductase inhibitors. HMG-CoA reductase inhibitors are competitive inhibitors of HMG-CoA because they are structural analogs to HMG-CoA. HMG-CoA is the substrate for HMG-CoA reductase, and statins like lovastatin, mavastatin, atorvastatin, most of the drugs which belong to statin category, so they end as statin, you can remember that way. So they are all competitive inhibitors of HMG-CoA reductase, and they are used in the treatment of hypercholesterolemia. So this is how a regulation of cholesterol in our cells is done in a variety of ways. So I have touched upon almost all ways of regulating cholesterol biosynthesis in our cells. I hope this video has helped you in understanding this very important concept in cholesterol biosynthesis, that is the regulation of cholesterol biosynthesis. Thank you.